So in this experiment we have taken a beaker in which we have filled the beaker with the water, and we have used a thistle funnel inside which we have put a strong sugar solution and covered it with the cellophane paper. Or you can use an egg membrane or animal bladder, and we have suspended it as it is shown in the figure. We have marked it with the initial level, and after some time we are seeing it that the level of this sugar solution is rising. Now let us see what are the major conclusions drawn from this experiment. The three main conclusions from this experiment are: the first one, in the experimental setup, some water of the beaker passed through the cellophane paper and entered into the thistle funnel containing the sugar solution. Sugar from the thistle funnel has not passed into the beaker. And the third one is the cellophane paper acted as the semi-permeable or differentially permeable membrane. It has allowed the water molecules to pass through it but not the sugar molecules. So this experiment suggests that the water from the beaker passed into the thistle funnel through the semi-permeable membrane, and it was found that osmosis took place. So this is an experiment related to the osmotic pressure. In the previous experiment we saw the same condition, but in this experiment we'll see that the thistle funnel has a piston over it and some weight is put over it. We'll see after some time that there is no entry of water inside this thistle funnel, and the sugar solution will not change its initial position and it will remain in the initial level. We'll see that this osmotic pressure is equal to the weight or the pressure required to nullify the osmosis. So therefore, what is osmotic pressure? Osmotic pressure is the minimum pressure that must be exerted to prevent the passage of the pure solvent into the solution when two are separated by a semi-permeable membrane. Here the semi-permeable membrane is the cellophane paper. Now let us see about another term, that is tonicity. What is tonicity? Tonicity is the relative concentration of the solutions which determine the direction and extent of diffusion. Based on this tonicity, the solutions are of three types: isotonic, hypotonic and hypertonic. Here three cells are placed in this solution. You can see the change in the plasma membrane and inside the cell content. The cell wall will remain the same, but the plasma membrane in three of them will be changing, except in this isotonic solution. Now let us see what is isotonic solution. The relative concentration of the water molecules and the solute on either side of the cell membrane is same, so there will be no change. The cell shape and the size will remain unchanged. That means it will be similar. Next is hypotonic solution, that is the solution outside the cell has lower solute concentration than the fluids inside the cells. That's why the water is getting inside the cell. Endoosmosis occurs as water molecules move into the cell. The cells shrink in size . It is happening in both, okay? So don't see this point, you just see this point, that there is the water enters into the cell. So the plant cell is placed in a hypotonic solution. Next one is hypertonic solution. The solution outside the cell is having the highest solute concentration than the fluids inside the cell. Exoosmosis occurs at the cell as the water molecules move out of the cell. So this is the condition. The plant cell placed in the hypertonic solution. You can see here the plasma membrane along with the cell contents, it just shrink, while here in the hypotonic solution it is getting elongated more and more. Now we'll talk about turgidity and flaccidity. Let us see what does turgidity and flaccidity means. When a cell reaches a state where it cannot accommodate any more water, that means it is fully distended, and the condition is known as turgidity. And flaccidity is the reverse condition of that is plasmolysis. When a cell is turgid, its water is stretched under the pressure from inside, and in its turn it presses the cell contents towards the center of the cell. The pressure of the cell contents on the cell wall is known as the Turger pressure, while the pressure exerted by the cell wall on the cell content is termed as the wall pressure. That is why you will find sometimes the fruits and vegetables are just bursting due to this wall pressure or the Turger pressure sometimes. This is a demonstration of the Turger pressure. These are the uses of turgidity to the plant. Builds up the root pressure in the root cells. Turger pressure helps the mushroom seedlings to sprout through the hard ground. It helps in Turger movement. Turger helps in opening and closing of the stomata and provides rigidity to the soft tissues. Next is plasmolysis and flaccidity. A fully distended plant cell which is kept in the salt solution for a few minutes, then it will lose all its descendant appearance and the cytoplasm will shrink. The plasma membrane will be withdrawn away from the cell wall, and the shrinkage of the cell wall is called as the plasmolysis, and the condition is known as flaccidity. The recovery or the reversal of the plasmolysis is known as the deplasmolysis. Sometimes we do the salting of meat, or addition of the salt to the prickles is a method of killing bacteria by plasmolysis. Through this, the bacteria's cytoplasm shrinks and all the water comes out of it. The water is drawn out from the bacterial cells, and thus the cells are killed. You can see how in the last one the cytoplasm is shrinked. Next is root pressure. It is a pressure that is developed in the roots due to the continued inward movement of water through cell-to-cell osmosis, which helps in ascent of sap through the stem. Here one experiment is given on the root pressure. We can see how root is giving its pressure. You can see here is the cell sap, and cell sap is continuously being taken, and what happens? This indicates that the mercury level is rising in this manometer. It shows that the root pressure is acting with the help of the ascent of the cell sap. Next comes the bleeding and guttation. Bleeding is sometimes not only human beings or the animals bleed, but also the plants also bleed. That is the loss of water through the cell sap through a cut stem. If sometimes we cut the stem, we see that there is loss of the water. Next, sometimes in the morning you can see that through the leaf veins or end of the leaf veins you will find certain tiny drop like structures on the margins or on the tips of the leaf. This is excessive loss of water through the guttation. Now let us see what is the importance of root hair and ascent of sap. Root hair contains cell sap which has higher concentration of salts as compared to the outside soil water. The different sets of osmosis, and thus the roots absorb the water. Absorption of mineral elements from the soil involves active transportation by students. You have learned earlier about xylem and phloem, and they together constitute as the vascular bundles. You know that xylem is responsible for conduction of water from the roots to all the parts of the plant, while phloem is responsible for providing the food from leaves to all the parts of the plant. Now let us see what are the forces that contributes to the ascent of sap. The first one is root pressure, second one is capillarity, next is transpirational pull, and last one is adhesion. Root pressure, it builds up sufficient force to push the sap in the xylem vessels up to a certain height. Next is capillarity. The narrow diameter of the xylem vessels causes the water to rise from a lower level to fill up the vacuum created by the loss of water due to transpiration. Then there is transpirational pull. Because water is lost by transpiration, so more and more water is pulled up due to this transpirational pull. And what happens? A large continuous column of water is produced throughout the stem. The last one is adhesion. That is, adhesion causes water to stick to the surface of surface of the cells and draws more water. This kind of force is important for all the trees. So these are the four forces which contributes the ascent of sap and allows the water to go up to heights of the plant. So this was all about this chapter. I'll send you a PDF. In that you will get the entire thing.